Today we will discuss the second lecture of the cell cycle. In the cell cycle, you understood that the first phase in the cell cycle is the interphase, which is divided into three subphases. That's the G1, S, and G2. Now begins the M phase. First, we will discuss the mitosis. The mitosis is the first M phase and it is equational division. Equational division means that at the end of the mitosis, from a single cell we will get two cells and both the cells will have the same chromosome number as is found in the parent cell, and the size of each cell would be equivalent to that of the parent cell. That's why we call mitosis also as equational division. The mitosis doesn't occur in germ cells, it occurs only in the general body cells, which we call as the somatic cells. The mitosis is a continuous process, but for the sake of understanding, it is divided into four phases. That's the prophase, metaphase, anaphase, and telophase. So first we will start prophase. Now in the interphase, the genetic material occurs in the form of chromatin. The chromatin is not the highly condensed and highly compacted form of the DNA. Now when the cell enters into the prophase, the first phase of the mitosis, the condensation of the chromosomal material begins. Now the process of chromosome making begins, but the chromosomes are not completely formed in the prophase, but they are in the process of formation. So the condensation of the chromosome material begins in the prophase. Then the centrioles, in case of animal cells which have already divided during the interphase, they will move towards their poles. And at the end of the prophase, you see there would be no Golgi apparatus, no endoplasmic reticulum, no nuclear membrane, and no nucleolus within the cell. This is the early prophase and this is the late prophase. In the late prophase, you will see in this diagram the nuclear membrane begins to disappear. The chromosomes are in the process of formation. When the prophase is over, the second phase starts, which we call as the metaphase. In the metaphase, the chromosomes get aligned at the equator. These are the poles and this is the equator. The chromosomes get arranged at the equator and they form the metaphase plate. Again, very important point regarding the metaphase is that the chromosomes are most clearly visible in the metaphase. If you want to understand the chromosomes or see the morphology of the chromosomes under electron microscope, it can be better understood during the metaphase. Each chromosome, when you view it in metaphase, it is composed of two chromatids which are attached to one another at the centromere. After that starts the anaphase. In the anaphase, the centromere splits. The centromere, it gets splitted during the anaphase, and these chromatids then move towards the poles. Poleward movement of the chromatids occurs during the anaphase. And after that, there is telophase. In the telophase, the chromosomes begin to decondense. This is the telophase. The chromosomes begin to decondense, and it is the process of now chromatin formation. And the Golgi apparatus, nuclear envelope, and nucleolus begin to reappear. And at the end of the telophase, now what happens? The nucleus has divided. Now the cytoplasm will again also divide, and that very process is known as the cytokinesis.